The true impact of the SARS-CoV-2 virus on the Spanish population has been a mystery. Over 28,000 people have died, but the government was unsure how far the virus had spread through the population. Now, a random study of 35,000 households across the country, testing over 61,000 people, has revealed only 5% of the population maintained antibodies to the virus. Professor Marina Poyan was the lead author.
And in those places more heavily heated, we have 10, 12%, 14% is the top. That means that we are very far away from the herd immunity that we are looking for, because specialists in herd immunity had estimated that in order to have this group immunity to prevent the spreading of the virus, we need 60% of people with antibodies.
Other experts have estimated that at least 70% of the population would have to recover from COVID-19 to halt the pandemic. But just because a person has antibodies, it does not mean they are protected, as immunologist Zania Stamataki explains.
We don't even know that the antibodies that people develop are protective. So this is something that still is out there, and if you have had coronavirus infection and you have detectable antibodies, we cannot say as scientists for sure that you're protected from reinfection and you're protected from severe symptoms. So we're still asking people to shield even if they have antibodies.
Antibodies are Y-shaped proteins which recognize other proteins called antigens. Antigens are molecular structures that may be present on the surface of a pathogen, such as a virus, which triggers an immune response when it enters the body. B cells have antibody receptors, which, if matched to the antigen, cause the B cell to bind itself to the antigen. Once bound, the antigen is ingested and broken down inside the B cell, with the resulting product placed on the surface of the B cell, known as major histocompatibility complex 2, or MHC2. Helper T cells bind to the MHC2 and release cytokines. The cytokines and T cell help cause the B cell to clone itself into memory B cells containing the same receptors, and also into plasma cells. The main function of plasma cells is to release antibodies. The antibodies created are specific to the antigen which was bound to the B cell in the first place. The memory B cells can last for years, and they can react rapidly in case of future reinfection. The study did not look at the neutralizing ability of the antibodies, but it did reveal that 14% of people who had mild symptoms and tested positive for antibodies in the first round of testing saw a decrease in the antibodies in tests done only a few weeks later. In a study by King's College London, the antibody levels of more than 90 patients and healthcare workers were monitored for 3 months after symptoms emerged. Tests revealed that while 60% of people had a potent antibody response at the height of the virus infection, only 17% retained that potency 3 months later. The study, that has yet to be peer-reviewed, found that antibody levels rose higher and lasted longer in patients who had severe cases. These results have implications for the development of a vaccine.
First of all, when you cleared the infection, was it your antibodies that did it? Are your antibodies sufficient to prevent you from having severe symptoms in the future? How much antibody is enough? And also the quality of those antibodies. So are these antibodies able to neutralize the virus from entering your cells? So the quality of the antibodies is important as well. To answer that question, you need to do laboratory-based tests, which are not readily available. The good news is that people that have had a lot of contact with infected individuals, so healthcare workers, for example, that may have had the virus and then they go back to work, we are not really seeing a lot of evidence of reinfection in these groups. So it is possible that they are protected. The data is a bit slim still for this, so we're waiting to get more good confirmatory assays.
As the world reopens, thorough testing of the population is crucial. For those who become infected but don't develop antibodies, the body has other defences. A Singapore study has highlighted the power of T cells, which are another immune cell. The research found SARS patients still retained their memory T cells 17 years after they were infected. So some virologists have suggested that, we're underplaying the importance of T cells, which can kill viruses. So are we focusing too heavily on antibodies?
We have found in patients that have recovered from COVID good T cell responses against the virus. So we know that the good news is you make antibodies and you make T cell responses. The next question is, how long do they last? Which one's better? We don't know. We don't know which one has contributed to clearing your virus. We don't really know that yet. Data show so far from other coronaviruses that your antibodies, for example, to SARS and MERS, they might wane after a couple of years, but T cell might last longer. So it is possible that you get a more long-term immunity from memory T cells that stay behind after your first infection, and they're able to act immediately following reinfection. For SARS-CoV-2, which causes the COVID-19, we don't know that yet, but these assays are now being done.